Bonjour tout le monde. Hello everybody. Bienvenue à cette conférence UCIAS. Je suis Thomas Bessen, je suis le directeur de l'UCIAS. The conference is going to be in English, but I think I'll do the introduction in French, or I'll start in French. Donc, Jean-Louis Mandel va présenter la première conférencière aujourd'hui, mais je voudrais d'abord vous parler un tout petit peu de l'UCIAS. Donc, l'UCIAS, c'est l'Institut avancé de l'Université de Strasbourg qui soutient des recherches aux frontières des connaissances les plus lointaines possibles et de préférence pluridisciplinaires. Et nous faisons ça depuis une douzaine d'années. Et nous avons, il y a 3 ans, commencé ce qu'on appelle des chaires par domaine de 2 ans, qui sont pour interagir plus fortement avec la communauté. Alors, il y a une chaire par domaine, les sciences sociales humaines, les sciences de la vie, et puis les sciences dites dures, physique, chimie, ingénierie, etc. Alors, la chaire de sciences de la vie s'appelle la chaire Paul Ehrlich, et on a le plaisir d'aujourd'hui d'avoir une nouvelle personne dans cette chaire, c'est Hélène Dollfuss, qui va être présentée bientôt par Jean-Louis Mandel. Alors, ces chaires sont... L'UCIAS s'est inspiré du Collège de France à Paris, qui est le plus vieux institut d'études avancées du monde. Hein, ce n'est pas celui de Princeton. Le Collège de France a presque 500 ans, a bientôt fêté ses 500 ans, et marche toujours très bien. Et donc, nous, on a une douzaine de chaires, ça fait une douzaine d'années qu'on existe, mais on espère aussi continuer pendant peut-être une centaine d'années. On ne sera plus là, mais ça sera très bien. Et dans le but, au Collège de France, tout le monde qui est nommé à une chaire, que ce soit d'une année, 2 années ou plus, ou 5 ans ou à vie, on font une leçon inaugurale, et on a décidé de faire la même chose il y a quelques années. Et donc, voilà, ça va être la 3e fois qu'on fait ça. Et la pression est sur Hélène, voilà, aujourd'hui. Alors, on a 2 conférencières, Hélène Dollfuss et ensuite Monica Monolescu, sur des thèmes totalement différents. Elle va être présentée par Roland Recht ensuite. Et donc, entre les deux, on a une petite pause café à l'extérieur. Voilà. Alors, la conférence d'Hélène va être en anglais, parce qu'il y a des gens qui ne sont pas francophones dans l'audience. Et ensuite, la conférence de Monica sera en français. Voilà. Et toutes les 2 conférences vont être enregistrées. Donc, je laisse la parole à Jean-Louis pour présenter Hélène Dollfuss.
Voilà, comme ça fait moitié-moitié. Et en vous priant de m'excuser de mon retard, je ne sais pas si c'est génétique ou si c'est multifactoriel, mais je n'arrive pas à en guérir. Enfin, je fais peut-être pas assez d'efforts. Voilà, donc je vous prie de m'excuser. Et c'est la dernière qui a été faite. Modifiez-le.
C'est celui-ci ?
Ah non, il faut attendez. Take, voilà, il faut faire comme ça. Ah. Voilà, chez UCIAS.
Ah, ça sent bien.
Voilà. Alors la dia est en anglais, donc parce que c'est juste pour rappeler qui est Paul Ehrlich, parce que c'est un nom, voilà, c'est un nom, il est sur des instituts, il a eu le prix Nobel, mais donc et si vous regardez Wikipédia, il y a des pages et des pages en anglais. J'ai pas essayé l'allemand, ça doit être pire. En français, c'est plus court. Alors j'ai un petit résumé de l'anglais, c'est juste pour rappeler que c'est un immense scientifique allemand, enfin médecin et scientifique allemand, qui a quand même un lien avec Strasbourg, puisqu'il a fait une partie de ses études de médecine à Strasbourg. Après, sa carrière, c'était surtout à Berlin, et c'est vraiment un pilier de la médecine scientifique et de la recherche médicale au début du XXe siècle. Parmi les découvertes qu'il a fait, alors il a développé toute une série de méthodologies qui a permis de mieux caractériser par des techniques de coloration les bactéries, les cellules sanguines, ce qui a permis effectivement d'étudier les leucémies, de débuter la compréhension de l'immunologie, etc. Il a eu le premier l'idée de faire du criblage pour des composés chimiques qui pourraient interférer, être des moyens thérapeutiques pour des maladies humaines, et notamment c'est lui qui a développé le premier médicament pour la syphilis, dont vous savez que jusqu'au début du XXe siècle, c'était aussi catastrophique, je dirais, que la tuberculose, ou à peu près. Et donc c'est le premier médicament qui a été, semble-t-il, aussi le médicament le plus vendu, parce que effectivement, avec d'ailleurs une opposition à un moment des corps médicaux en disant si on a un médicament, ça va augmenter les turpitudes des gens parce qu'ils auront plus peur de ce qui permettrait de garder la moralité. Bon. Et donc aussi, c'est un des créateurs de l'immunologie. Donc voilà, c'est sous ces auspices qu'on a cette chaire, dont la première titulaire a été professeur Brigitte Kieffer dans le domaine des neurosciences. Voilà, alors maintenant on va passer à Hélène Dollfus, que je suis très heureux de présenter. On se connaît, je pense, depuis à peu près ton retour à Strasbourg en 2003, quelque chose comme ça. 98. Comment 98. Ah, bah, décidément. Oui. Ok. Ah oui, d'accord. Ok. Et donc, on peut passer la prochaine. Hein, où est-ce que c'est moi? Ah, c'est ça.
Je pense que c'est ça.
Voilà. Alors, j'ai voulu mettre un peu de détails parce que justement, la carrière, au départ, par l'internat de médecine à Strasbourg d'Hélène Dollfus, était, où elle s'est vite spécialisée en ophtalmo, mais a pris un tournant que trop peu d'internes prennent, hein, c'était assez exceptionnel, puisque, je crois presque la deuxième année d'internat, elle est allée à Paris, et à Paris, elle a eu une année recherche, ou même deux, non? C'est deux. Voilà. Deux années recherche, et puis dans un excellent endroit, puisque c'était à l'hôpital Necker Enfants Malades, dans le laboratoire d'Arnold Munich, qui était un des laboratoires phares dans l'étude des maladies rares génétiques. Et là, elle a commencé à travailler sur les maladies rares génétiques de l'œil, donc responsable de cécité, maladie de Leber, il y avait différents types de maladies de Leber, syndrome de HR, etc. Et avec, comme jeune interne, tout de suite, toute une série de publications dans cet endroit extrêmement actif. Et puis, après, elle est allée faire une année dans un célèbre institut d'ophtalmologie à Londres. Donc cet aspect de commencer très tôt dans les études d'interne, enfin dans ces fonctions d'interne, de la recherche de haut niveau et de la recherche internationale de haut niveau, je crois que ça c'est déjà montre tout à fait l'originalité de son parcours. Elle est revenue à Paris d'abord, où elle a été chef de clinique dans le service d'ophtalmologie dirigé par le professeur Duffier, donc, qui était un des grands patrons de l'ophtalmologie, mais tout à côté, effectivement, aussi du laboratoire d'Arnold Munich, et donc de tout ce qui bouillait sur la génétique là-bas. Donc, à la fin de son clinica à Paris, elle revient à Strasbourg, et là, elle prend la décision de continuer à travailler sur les maladies ophtalmologiques, mais pas que, et donc d'aller vers la génétique médicale, vraiment. Et donc, elle a eu un poste de MCUPH en génétique médicale. Et puis, au bout de quelques années, donc... ah oui, effectivement, donc c'est en 97 que tu étais revenu, donc c'est là qu'on s'est connu. Et puis, en 2003, elle devient professeure de génétique médicale à Strasbourg. Et puis, elle a été très active, et je pense qu'il faut souligner, avec des fonctions à la fois recherche et recherche dans le domaine aussi du soin. Alors, j'ai marqué, parce que c'est très, très impressionnant, donc elle dirige un service de génétique médicale des US, et la génétique médicale a vécu ces dernières années une véritable explosion grâce au progrès, je dirais, des possibilités de séquençage et de la connaissance d'un nombre très rapidement croissant de maladies. Ça étonne toujours les gens de se dire qu'on découvre de nouvelles maladies, je dirais presque chaque semaine. Alors, mais ces maladies, elles devaient exister avant, sauf qu'on n'arrivait pas à les identifier. Par exemple, ce qu'on appelait la rétinite pigmentaire, c'est maintenant quoi, 150 maladies différentes? Oui, et parce que tout d'un coup on trouve que chez tel ou tel malade, il y a un gène dont on ne savait pas qu'il était impliqué, et puis une fois qu'on a chez quelques malades, on s'aperçoit qu'en cherchant, il y a pas mal de malades qui sont atteints de rétinite pigmentaire à cause d'une mutation dans ce gène particulier. Donc on découvre de nouvelles maladies, donc beaucoup d'activités. Et puis au début, la génétique, c'était le domaine un peu quand même de la pédiatrie, donc des maladies de l'enfant. Et on s'aperçoit maintenant qu'il y a des maladies génétiques qui se déclenchent à l'âge de 50 ou 60 ans, et qui sont tout à fait des maladies génétiques. Donc là, ce service de génétique médicale, mais l'explosion de la génétique et le fait qu'il y ait, je dirais, des associations de malades, et notamment l'AFM Téléthon, qui a beaucoup poussé à ce que se développe les moyens de suivre, de diagnostiquer, de traiter, si possible, les malades atteints de maladies génétiques. Il y a eu une organisation, au départ assez originale en France, de centres de référence pour que ces malades où souvent les médecins disaient on ne connaît pas, c'est rare, donc qu'il y ait des centres d'expertise maladies rares dans toute la France, structurés en centres de référence, centres de compétences. Et donc Hélène a été tout de suite partante dans ce domaine et a créé un centre labellisé, donc centre de référence sur les maladies rares de l'œil, qui s'appelle le Cargo. Alors ça, c'était à Strasbourg. Et puis, dans ces plans maladies rares, on a structuré les différents centres de référence maladies rares en filières qui rassemblent les centres de référence pour certains types de maladies. Et donc, Hélène est devenue coordinatrice nationale de la filière maladies rares sans gènes, qui, comme son nom l'indique, c'est les maladies génétiques des sens, c'est-à-dire surtout, évidemment, l'œil, mais aussi l'audition, l'oreille. Donc, c'est une très grosse responsabilité. Et puis, comme l'exemple français, pour une fois, on peut faire un petit cocorico, pas toujours, même en génétique, mais là, l'exemple des centres de référence a fait réfléchir la communauté européenne, et il y a eu des centres ils ont décidé de faire des networks de centres de référence pour les maladies rares. Et Hélène est devenue coordinatrice de RNAi. Ça, je pense que c'est le problème quand on fait quelque chose de trop bien. Les gens se disent, puisque c'est bien, elle peut faire au niveau encore plus, qui peut poser de temps en temps des problèmes. Donc ça, c'est ce qui est sur l'organisation des soins, mais aussi la recherche, parce que sur toutes ces nouvelles maladies, il faut les comprendre, il faut savoir comment au mieux les prendre en charge et les traiter, essayer de structurer pour qu'il y ait des nouvelles thérapeutiques qui puissent être testées. Donc tout ça, c'est dans le soin, mais dans le soin recherche. Et puis, elle dirige aussi, ou elle a créé plutôt, une unité INSERM. Elle a été nommée à l'Institut universitaire de France en 2014, qui montre qu'elle est tout à fait reconnue. Et puis, elle a eu aussi le prix recherche INSERM il y a quelques années, donc c'est quelqu'un qui est très reconnu. Alors, encore 2 minutes. La prochaine dia, parce que moi j'ai... ah oui, c'est moi, décidément. Voilà, c'est une dia de Hélène, une ancienne, je pense que tu les as un peu modifiées depuis. Mais ce que je me souviens de manière très, très nette, c'était... on avait un groupe de génétique de l'Est où on faisait des réunions. Et un jour, Hélène a parlé, je pense que c'était peu après que tu sois revenu à Strasbourg, d'une maladie dont j'avais, je pense, jamais entendu parler, ou alors juste un nom comme ça, mais je ne savais pas ce que c'était, qui était le syndrome de Bardet-Biedl, qui était une maladie d'hérédité récessive, donc on se disait c'était une maladie. Surtout qu'elle était tellement particulière, parce que c'était une maladie où vous aviez... je dirais malheureusement évidemment pour les patients, des choses dont on se dit que ça n'a pas grand-chose à voir entre eux. Bon, il y avait des problèmes de vision, donc ça c'est de rétinite. C'est pour ça qu'au départ, je pense, Hélène s'y est intéressée. mais il y a souvent une obésité qui peut être une obésité massive. Il y a des caractéristiques squelettiques telles que des polydactylies aux pieds ou aux mains, des anomalies rénales qui peuvent être source de déficience rénale sévère, des problèmes d'audition, hypogonadisme, enfin, des affections cognitives, pas trop sévères en général, mais enfin voilà. Donc, tout ça, on peut se dire, qu'est-ce qu'il y a à voir ? Et moi, je sais qu'en l'écoutant, je me dis, ça doit être un facteur de transcription qui régule l'expression de gènes dans ces organes. Il doit y avoir un gène, un. Et puis en 2001, on trouve un gène. Et puis Hélène Dollfuss, qui avait constitué des cohortes de patients, en 2006, identifie un gène, puis un autre, puis un troisième après, etc. Et à l'heure actuelle, on doit être à peu près 20 ou 26. Et en fait, ceci a ouvert un nouveau champ non seulement de la médecine, de la biologie cellulaire. Et je crois que je vais te laisser parler du cil pigmentaire, mais c'est juste, voilà, ça c'est de la bibliométrie. De temps en temps, ça sert à la bibliométrie. En haut, c'est le nombre d'articles et le nombre de citations à ces articles sur Bardébil. Bardébil, c'est connu depuis le début du XXe siècle. Vous voyez qu'il y avait quasiment une publication par an, ou tous les 2 ans, etc. Et puis, quand la génétique commence à bouger, vers 90, ça bouge un peu. Et puis, c'est l'explosion à partir de 2000-2005. Et à côté, vous avez quelque chose de biologie cellulaire. Le cil primaire en dessous. Cil primaire, c'était une observation de microscopiste électronicien. Personne ne savait ce que c'était. Et donc, personne ne travaillait dessus. Et tout d'un coup, grâce à des maladies telles que Bardébil, telles que la polykystose rénale, tout d'un coup, on fait le lien entre ces pathologies et le cil primaire. Et le cil primaire devient un sujet absolument énorme. Voilà. Et je vais lui laisser. Okay. Je te laisse 10 minutes.
Merci beaucoup Jean-Louis pour cette introduction. And so I'm going to switch to English and welcome everybody to this inaugural Paul Ehrlich conference. So it's my great honor and my great pleasure to be here and to occupy this position for 2 years. Jean-Louis has recapitulated the career of Paul Ehrlich, but he was a tremendous scientist, a tremendous MD, and one of the first to discover a synthetic drug for syphilis. And I hope I hope he would have been interested to listen about rare diseases and ciliopathy. So it's a real honor, and I really thank the USIAS committee for this nomination. So today I will go through rare diseases overall, and I will focus on ciliopathies and take the example of ciliopathies to enhance and underline very specific problematics that we encounter in rare diseases. So first of all, what's a rare disease? It affects fewer than one people over 2,000. There may be ultra-rare diseases, which are even rarer, but it is a public health issue because more than 30 million people in Europe are affected. That means 4% of the population during the lifetime will be affected by a rare disorder. There are more than 6,000 rare diseases, and more than 3/4 are of genetic origin. And a lot of these, they start in childhood and they last during the whole year. They reduce the quality of life and sometimes the life expectancies. One problem is that most of the diseases are underdiagnosed, and it takes more than 5 years to get, in average, to get a diagnosis. It can be much more. And another point is that more than 95% are still devoid of a specific treatment. So it's a huge area of medicine where we have to find specific treatments for the patients. And there are only today, 250 orphan medicines. So there are huge challenges in rare diseases. First, at the clinical level, natural history, knowing more about the natural history of these disorders. There are new disorders we didn't know about before, still being discovered. Understand the genetic causes, and there are still patients for which we do not find the genetic cause. Understand the biological mechanism, and this is very important because it can unveil new biological actors, and this is very important to understand the pathogenesis and to pinpoint specific targets for therapies, for instance, and also how rare diseases can really enlighten common diseases. And rare diseases may really serve common diseases, as I will try to show you during this talk. And I will focus on my field, which is the one of the fields, which is ciliopathies, as an example for rare diseases, and I will emphasize each time how it enlightens more or less the whole field of rare diseases. So ciliopathies, there are more than 1,000 cilia-related genes, more than 35 clinical entities, different syndromes, and many patients worldwide. So what we apply, our philosophy, I would say, in our research is what I call the translational genetic rare disease research. We always start from patients, we identify the genetic cause, then we try to understand the mechanism with the basic and applied research. This is to come back to therapy and to come back to the patient. So it's a virtuous circle. I will illustrate here today now with ciliopathies. So what is a cilia, and where do these organelles come from? Very interestingly, they were first discovered by a very prolific professor in the Netherlands, Professor van Leeuwenbeek. I hope I pronounce rightly, but he was extraordinary because, very interestingly, he was working on textile and inspecting the quality of the textile with lenses. And this prompted him to develop different lenses and have immobile lenses. And finally, he discovered microscopy, and he discovered unicellular organisms. And he also noticed that these animalcules, what he called animalcules, had strange little feet or legs. And this was the first description of flagella. He also described in human spermatozoids. So he really was the first to discover these cilium. And then came very important the discoveries by Paul Langerhans and also Professor Zimmermann in the University of Bern, where they observed with the new microscopes and drawed cilia cells from different organs like the kidney, like the thyroid, and they really identified a protrusion that was like a cilia, and what was called by Zimmermann the central Geisen, and this was the central flagella. So, and actually, Zimmermann reported this for the first time in Strasbourg in a meeting, but there was no abstract at the time, but it was reported for the first time in Strasbourg in 1894. So, this was really very repeatedly reported. However, for years and years, more than a century, as Jean-Loup just cited, it was a complete lack of totally neglected organelle. There was no publications, except for when the electron microscopy, the transmission electron microscopy was used. Then there was a description of the structure of the bases, for instance, of the connecting cilium, etc. But it was really, at the level of PubMed, totally flat. So and this organelle was considered as vestigial. However, the third millennium is really the golden age of this organelle, and it started with with a biflagellate organism, Chlamydomonas, where a mutant, EFT88, which is a mutant that is important for a gene that is important for the flagellum, for the transport in the flagellum. When this gene was mutated, there were absent flagella. Interestingly, a mouse model with the same gene mutated showed cystic disease in the kidney. And that's where the switch was really done, where it was understood that the primary cilium could have an effect at the level of an organ, and also if it's absent or doesn't function well, may be at the origin of clinical symptomatology. Oops, sorry. So I will go very quickly through this, but in the human body there are two types of cilia. The motile cilia we have at the level of the airways, and these are very numerous at the level of one cell. And in the other type of cilia is the primary cilia, that is non-motile, present in one exemplar in each cell in almost all cells of our body. And for the motile cilia, there are also some conditions, primary ciliary dyskinesia. I will not get into these diseases today. But for the primary cilium, accordingly to where the organ, where the protein, the specific cilia is implied, you have different organs that are affected. As the primary cilia is present in almost all cells of the body, you can imagine that a defect may lead to different clinical manifestations. And these are listed on the right of the slide. And also, it was discovered that a number of rare syndromes which had these different manifestations at the level of the kidney, at the level of the central nervous system, the skeleton, also the retina, it was discovered that rare syndromes were linked to primary cilium, and this is what I'm going to demonstrate now. So some of these syndromes are cited here: the Bardet-Biedl syndrome and the Alström syndrome. Alström syndrome, I will not get into this today. I will mostly focus on Bardet-Biedl syndrome. What is this condition? It was described by Georges Bardet in 1920 and Arthur Biedl in 1922. Very bright clinicians, because they understood that in the same family, two siblings presented a very specific set of symptoms, meaning, and these are obesity, polydactyly, an extra digit, also kidney problems, sometimes some intellectual deficiency. And Bardet and Biedl really described for the first time this syndrome that leads to multiple handicap. These children get blind when they are very young. At 5 years of age, most of these children are blind, and the obesity is a huge problem during their whole lifetime. So we got interested into this syndrome, as Jean-Louis mentioned, in the years 2000. And the first description of the relationship between this syndrome and cilia was done in 2003 with the description of one of these genes. And it became very clear that genes encoding for proteins important for the primary cilia may lead to different clinical manifestations. Here, obesity, kidney dysfunction, and retinal degeneration. And this is the beginning of a very long story that is still ongoing. So we will focus on Bardet-Biedl syndrome and take again this virtuous circle to get through the different aspects of this syndrome and of primary cilia biology. So first of all, patients. We start always by patients in our research, and cohorts of patients, it can be one patient or hundreds of patients, is extremely important in medical genetics and in rare diseases. So, for instance, for Bardet-Biedl syndrome and Alström syndrome, which shares a number of features with Bardet-Biedl syndrome, natural history studies are really important. And this is the case for all rare diseases. because we do not know enough about the progression of different organ manifestations. It has been neglected, natural history, for a number of years, but now it's coming back also with the artificial intelligence tools. And what is very important is to know about the progression of diseases, for instance, when you develop drugs for clinical trials. So in Strasbourg, we have developed the COBALT cohort for studying the natural history of Bardet-Biedl syndrome. We have more than 200 patients we are following up. At the same time, it is very important to diagnose these patients. I was telling you that it can take a very long time for any doctor to pinpoint that it is a Bardet-Biedl syndrome. So we have revised, and thanks to the introduction of Jean-Louis, you know now that there are European networks for rare diseases. And based on a collaboration with the European Network for Rare Eye Diseases, for kidney diseases, for endocrine diseases, and for developmental diseases, we have revised the clinical criteria and follow-up processes for this patient this year, taking into account the molecular level that was up to now not a criteria, but that appeared to be very important for the holistic care and follow-up of and diagnosis of these patients. So if we look, the next step is the molecular identification and the identification of new genes or new molecular diagnoses. This is a very busy slide, but it finally recapitulates the history of sequencing tools for rare diseases. And taking the example of Bardet-Biedl syndrome, we can see now that today there are more than 26 genes that have been identified. We have identified quite a few in our lab. And we can see that the technology has evolved. For the ones who know about Sanger sequencing, that was extremely tedious. Extremely precise too, but very long. And today we have next generation sequencing with whole genome sequencing. There has been huge progresses that has really helped to disentangle and understand all the genes that are not all today, but there are still a lot to discover, but to help to discover all genes implied in rare diseases and also in Bardet-Biedl syndrome. And for instance, today in our lab, and I'm happy to see that Sylvie Friand and Séverine Baer are in the room, we are still working on new genes. The challenges are different. Oops, I don't know what happened. Yeah, the challenges are different today because we are working sometimes on a very low number of patients, sometimes unique patients, and functional validations can be extremely challenging. So we are still working on novel genes. So identifying new genes, this authorizes us to transfer this to the hospital, to the diagnostic lab. And in Strasbourg, the diagnostic lab is very much, with Jean Muller, specialized in Bardet-Biedl syndrome. And we know there are 3 major genes. We know that the whole spectrum of mutations. In 90% of the cases, we are able to deliver a diagnosis, which is a real progress since the last year for these patients. And we are also able now, because of these new sequencing technologies, to solve cases that were unsolved, for instance, with very specific mutations like retrotransposon insertions that have been recurrently described in the literature now. So using these new sequencing tools also authorizes us to discover new mutational new types of mutations. Why is this important? Because from there, we can already return to the patient. Knowing the genes, we can propose genetic counseling and family planning. And this is very important for the patient. So this is the first added value of our research to help patients. So we consult the patient as an autosomal recessive condition, and it's impossible not to mention, if we are speaking about Bardet-Biedl syndrome, a very important paper in 2001 by Nicolas Katsanis about oligogenic inheritance. Although this was a notion that we challenged with Jean-Louis a lot, but it opened the door to what we call genetic modifiers that may be not only the two major genes that are necessary for an autosomal recessive condition and that will induce the disease. You can have a third mutation in another gene that can modify the phenotype and that can have an action on the severity, for instance, of the condition. So I will not get into the details of phenotype-genotype correlation, but it's a whole field in rare diseases to understand what are the factors that can really modify the condition. And for instance, for Bardet-Biedl syndrome, there are some genes. We know that the phenotype is less severe than other genes. We have also some very specific phenotype-genotype correlations. For instance, for a gene I will talk about at the end of my talk, LZTLFL1. And today there are a number of groups that are working on how to disentangle other ciliary genes in which variants could modify the phenotypes in these different conditions. So I will not get into the details, but it can be by using zebrafish or meta-analysis or genome sequencing. And looking at genome sequencing of all our patients, this is a problem because we need a lot of patients, and these are rare diseases, but we are getting there with international collaborations. The last point on this molecular genetic part is about all the genes have the same epidemiology worldwide. For us in France and in Europe, it's BBS 1, 10, and 12. But in other parts of the world, the epidemiology is quite different. This is important because when you try to find different therapies, like gene replacement therapies, this is gene specific. So that means that it can be treatment that can be active for some populations, but not for others. So this is an important work that is ongoing. So the second part is about understanding the mechanism underlying these conditions. And there has been a lot of research. Many groups worldwide are working on primary cilium and on other rare diseases to understand and what is the pathogenesis underlying the clinical manifestations. And for this, there are many tools that are used: in vitro tools, in silico tools, in vivo and ex vivo tools. So to understand Bardet-Biedl syndrome, the first very important discovery done by Nashuri was to understand that 8 of these BBS proteins, they assemble to form a complex that is called the BBSosome complex. So this was a very important step, that they are not acting independently, they act together for 8 of them. And their assembly is facilitated by a chaperonin complex with BBS 6, 10, and 12. And then there are other BBS proteins that are also very important for the functioning and the development of the primary cilium. And the different BBS proteins may act at different levels. For instance, trafficking proteins towards the base of the cilium, then helping the traffic inside the cilium. And this is very important because there is what we call intraflagellar transport, anterograde and retrograde. And for all this, there are many proteins implied, including BBS proteins. And probably this is the most important slide of my talk, because this really pinpoints why primary cilium are so central in biology. In fact, you should imagine that the primary cilia at the level of one cell is a cellular protrusion, and it's like a signaling antenna. And in the years 2000 also, it was shown that very important biological pathways are linked to the primary cilia. So most of you probably know about the sonic hedgehog pathways. Well, the receptor, all the trafficking is linked to the primary cilia. So we are here in a sort of communication of the cell with the environment. It can be a chemical stimuli, It can be a mechanic stimuli, like in the kidney, for instance, the fluid of the urine, that will activate receptors specifically present at the level of the membrane of the antenna. And then there is all the traffic going on in the primary cilia, and then it will activate in response the GPCR, and for instance with the second messengers that can be AMP, cyclic AMP, and it can activate the expression of genes to react to the external stimuli. So this is very important because probably the primary cilia has different activities at different levels of different organs and at different times. It can be important for the development of the cell, for the development of the organ, but also for its maintenance and its functioning. So this explains the syndromes, the clinical manifestations, but we still have a lot to learn. And for instance, the baby exosome was supposed to be only placed at the base of the cilium, but it has been shown that it actively participates to the intraflagellar transport in many ways. And also on the right side of the slide, you can see here the junction with the receptors. It's a hypothesis, but the junction with the receptors and the activation of the transport with the signaling pathways. And this is very important because this may be a therapeutic target. Another part that is emerging now, a lot of you probably have heard about extracellular vesicles. It's very popular, for instance, in oncology. Now it has been shown in a model of Bardet-Biedl syndrome mouse by a German group in Tübingen that the primary cilia is also active in releasing extracellular vesicle. So this is a field that is actually moving very quickly at the time being, and they showed it was linked with the Wnt signaling pathway. So coming back to our Bardet-Biedl and to our laboratory, of course we have been trying to model the condition. And this is the case for all rare diseases, but here we focus on ciliopathies and Bardet-Biedl. So for this specific field, there are a few different ways of modeling. One way we use very much is the skin fibroblasts of the patient. So very easily we can do a biopsy, a very small biopsy at the level of the patient. It's really nothing. For children, it's very, very much okay. And then we put in culture the cells, and we can look at the cilia of the fibroblast, and we can quantify the length, the morphology, and also test various signaling pathways like sonic hedgehog, for instance. This is in vitro. We are also developing, and I'll come back to that in a minute, organoids from patients affected by ciliopathies to study the retina and the kidney. And in vivo, there are many models. Zebrafish, we have collaborated with the KIT in Karlsruhe a lot for this, but also mice, and we have a number of mice in our lab we can use. And also, there has been described a natural model of monkey, which are mutated with symptoms in BBS7. So we use skin fibroblasts, and this is a complicated, busy slide, but we look at how the fibroblast, the ciliogenesis, that means the number of ciliated cells. We look at the length of the cilia, the phenotype also of the different signaling pathways, and the cell proliferation. This is tools we use every day to either characterize mutations or characterize new genes. But we don't only use skin fibroblasts. We also, we also use, we can also use cells that are in the urine, and for this we collaborate with Paris, with the Institut Imagine and Sophie Saunier. We collect urine of the patients, and then in the urine there are cells that are called the URECs, the urine renal epithelial cells. We can culture, and even Sophie Saunier, she's actually doing organoids from these cells. So, for instance, for studying kidney manifestations, it's also possible to use other cells. So if we continue on our pathway, I will focus on 2 clinical manifestations for the sake of time to try to understand why the symptoms develop and how we can act so they do not develop. First of all, the retinal degeneration. Why is there retinal degeneration in ciliopathies? The photoreceptor cells, the cones and the rods, which are very active at the external part of the retina for the light transduction. The light is transduced in an electric signal at the level of these cells. They are modified cilia cells. They have 2 parts, the outer segment and the inner segment, that is, and the junction of which has a structure of a primary cilia. It's the connecting cilium. And in fact, in many ciliopathies, you have a retinal degeneration. So why is there retinal degeneration? There are many hypotheses and still many probably to come. The first hypothesis, and we were working on this also in our laboratory, is that the transport is blocked. So from the external segment to the inner segment, from the outer segment, where the proteins are synthesized, to the outer segment where the phototransduction takes place, we may think that, for instance, rhodopsin, all these proteins are stuck in the inner segment, and then this induces a protein overload, unfolded protein response, and apoptosis. This is one hypothesis. But moving forward, it has also been discovered, and I explained to you that the babysosome was active in bilateral transport. Another hypothesis is that the protein also circulates in the external segment and cannot come back in the inner segment to be degraded. So there are many hypotheses, and I think we are still not through because the pigment epithelium has been also overlooked. So today there may be different ways of acting on the retinal dystrophy. To study this, we also use mice. And the Bardet-Biedl mice, they become obese, as you can see on the slide. And for the retina, you can look at the histology and also have electrophysiology. And this can be very good biomarkers. So the mice have been very much used also to develop pharmacological therapies and also gene therapy. With, for instance, an American group that has developed subretinal gene therapy for the Bardet-Biedl mouse. However, it's very difficult for many reasons. First of all, these mice are very fragile. Although they are obese when they are born, they are very small and they have a tendency to die very early on. So they are extremely fragile, so it's very difficult to breed them. This is one difficulty. The other thing is that it does not, for instance, at the level of the retina, totally recapitulate the human retina. And speaking of gene therapy, at the time being, there are a lot of discussions on should we really continue specific gene therapy. Generally speaking, we can speak of that in the discussion for some rare diseases where it's extremely heterogeneous, where there are a lot of different genes, and finding the right vector is also a huge challenge today. So, are there other models? Yes. So we are developing, like others, of course, iPS and organoid models. You know that this has been a revolution in science and medicine with the Yamanaka factors and the possibility to generate from a somatic cell a pluripotent cell using the Yamanaka transcription factors brought by a virus in the cells. And then once you have these pluripotent cells, you can differentiate them into different lineages, and thanks to very elaborated free extracellular matrix and 3D possibilities of culturing cells to develop organoids of many organs. So this is many organs. So this is a real revolution. You can have cardiomyocytes, you can have intestines, and also eye. So these organoids are, of course, extremely useful for medicine. It's a huge area of medicine, very difficult. I will tell a word of this in a minute. But it can be used, for instance, for drug screening, also for regenerative medicine. But we use it really to understand the pathogenesis. So we use it from the fibroblast. I was talking about the skin biopsy we do regularly for our patients. So we can also use these cells to bring them to be iPS cells, to reprogram them, and then to differentiate them into different organs. So with Paris, we work on kidney. And in Strasbourg, we work on the eye. And this is a very, very long process, which takes a lot of time. I'll come back to this in a minute. And what we want to use these retinal organoids for is to understand the mechanisms with, for instance, RNA-seq, etc., or the omics we can do. And we also look at these organoids for pharmacological targets, and also we can use them for validating, for instance, very difficult, what we call variants, genetic variants of unknown significance. So we are doing this thanks to a very precious collaboration with the Institut de Neurosciences de Montpellier, with Dr. Kalatsis. She is really training us. It's a fantastic collaboration. Our plan is to have organoids for all the major BBS genes from patients with classical phenotypes and with extreme phenotypes. We are very lucky because we have a full and a long-standing cohort, the COBALT cohort I told you about. We have patients with extreme phenotypes, and these extreme phenotypes hopefully will enlighten us on modifiers or specific targets for therapy. So I also put it the minimum time needed to generate these organoids from the skin biopsy of the patients until we have the organoid in the lab. It's more than 1.5 years. And it's extremely tedious. If you have a mycoplasma contamination, you can throw everything and start everything again. It's a bit like le mythe de Sisyphe, I would say. But we are getting there. I would have loved to show results, but we are still in the process of generating them, and we are obtaining our first ones now. So each organ is a challenge, and I will finish on the description of the organs before my conclusion on the obesity. So it's very strange to see that in these ciliopathies, the patients may develop obesity. Why is that? In fact, there are two hypotheses. I think one slide is missing, but never mind. There are two hypotheses. Either there is a problem at the level of the adipose tissue itself, the adipocyte, and the cells that will differentiate in adipocytes, or there is a problem at the level of the brain, the hypothalamus, where we know that there are cells that regulate appetite. And this is very famous pathways with the leptin and ghrelin that have been discovered a long time ago. And what is interesting is that in the brain, the hypothalamic brain, like a lot of neurons, they have a cilia, and they have receptors at the level of their cilia. I will come back to this in a minute. So today there are two hypotheses about the origin of obesity in ciliopathies. The adipocyte and the pre-adipocyte mesenchymal cells alteration. We have shown this a long time ago that the pre-adipocytes are ciliated. They were not supposed to be ciliated, but they are. And very recently, some other labs have shown that there is a link between the adipocyte and the signaling pathways of the primary cilium. And on the other side, and probably it's dual, peripheral and central, you have the central control of the appetite, with a lot of different models that have been generated, including organoid cells, neurogenous progenitors, and mice. Why am I speaking to you about this? Because what is important is that there is a drug that has been developed to fight against the obesity in Bardet-Biedl syndrome. So that's why I wanted to bring you here. And at the level of the hypothalamus, you have different receptors for instance, for the leptin, and it is known that there are other rare diseases in which you have a major obesity because these receptors, the genes encoding for these receptors, are mutated. So it has been shown that acting downstream with an activator of MC4R enables to correct the phenotype of these patients with a genetic obesity because they diminish their appetite and they diminish their food intake and their energy expenditure. It has been shown that the hypothalamic cells oscillated and also that the Bardet-Biedl proteins have an action at the level of the same cells as these genetic obesities I was just describing. So it was thought that we could repurpose the drug that was used for the severe genetic obesities for Bardet-Biedl syndrome and other obesities using this downstream receptor to activate more this downstream receptor, diminish the hyperphagia of the patients, and by then diminishing the obesity. So it has been proven, and using what is called the setmelanotide, which is a MC4R agonism, it indeed in Bardet-Biedl restores the signal at the hypothalamic level and diminish the hyperphagia and the obesity and metabolic consequences of the obesity. So this is to highlight that in rare diseases you can have trials, and we actually participated in Strasbourg with our COBALT cohort to the clinical trial that led the setmelanotide to be recognized by FDA and EMA. And interestingly, some patients are responders, other patients are not responders. But the patients who are responders, I can tell you it's a life-changing molecule because when you weigh 150 kilos, you lose 30 kilos, you get out of your wheelchair, you can go with your mother to the supermarket, you can get outside, and this is a real life-changing molecule for some patients. So there has been approval in the United States and in Europe. Is this always easy in rare diseases? No, they were very lucky because the industry, the pharmaceutical firm, had a big experience in rare diseases and in clinical trials. I can tell you that today, when it's ultra rare conditions, it's very difficult to get through phase 3 and phase 4 trials and to get the approval. But here, the drug is on the market in different European countries and helping a lot of patients. So I'm coming to my conclusion. So I showed you an example of translational genetics using the example of the ciliopathies and Bardet-Biedl syndrome. And this brings me to a few general comments. I have 3 before my take-home message. My first comment is about the therapy challenges in rare diseases. Today, we have a whole arsenal developed by many labs in the whole world: gene replacement therapies, gene editing therapies, antisense oligonucleotide therapies, cellular therapies, and also pharmacological therapies. So there are a lot of tools out there. This is the arsenal, and we would like to use this for all our rare disease patients. The reality is a bit different, and I just wanted to pinpoint on this because the costs are huge. The market of rare diseases and ultra-rare diseases is not helping because the number of patients are often very low. And actually, at the time being, we are seeing very sadly some lack of interest of some industries and investors that are retrieving their participation to trials in rare diseases. So this is going to be a big problem, especially for gene therapy, gene replacement therapy, where in conditions where there are more than 300 genes, or here 26 genes, addressing one gene, finally, it's a very low amount of patients. We can speak of that later on. And So gene agnostic therapies are preferred at the time being. This is the new wave coming up. And also how to deliver the drugs is also a big challenge, especially, for instance, for the retina. So we, in our lab, we are working with a pharmaceutical company to develop gene-agnostic therapies in ciliopathies. My second comment is about all the literature, the huge amount of literature coming out about ciliary, the primary cilium, in aging, for instance, and senescence, how you need to have a healthy cilium. Also, in neurology, for instance, there is a lot of work in Parkinson diseases and other common neurological diseases. And it is true that the cilia in neurons have been quite neglected, and they are very much present in neurons, but because the access to the cilia, to the study of the cilia was different from the other cells, it is a field that is really exploding. But I think we have understood that the cilia, the primary cilia, has a central role in biology, and is studied in a huge amount of fields in medicine, metabolism, hypoxia, also tumorigenesis. Of course, I didn't speak about that here, neurodegeneration. It's just the big question. I think not all these diseases are ciliopathies. There is a sort of tendency of extrapolation. Is it a primary or secondary effect of in these conditions? This has to be still studied and to see what is really defined as a ciliopathy. And my third comment, this one is for Jean-Louis Mandel specifically. I hope it's food for the brain. So I described to you in the phenotype-genotype correlation. I told you, remember this gene, the LZTFL1 BBS17 gene. This is a gene very well known in the trafficking of the BBSosome and the signaling pathway of sonic hedgehog. It was very well known. And we discovered in 2012 two families in Strasbourg. They were French families, one of Strasbourg and one from another part of France, who had a specific phenotype. They had the sixth digit was not on the side of the hand, but it was in the middle of the hand. Very, very special. And we discovered that these two families were carrying mutations in this gene. So we published, we had enough amount of functional data, and it was published, but there was no other family discovered until 2021. So we were a bit wondering, is it really a gene or not? And then now there is a fourth family that has been discovered. So it's ultra rare, ultra specific genotype-phenotype correlation of these families we have been following for a number of years. So what happened in 2020? Well, in 2020 there was the COVID pandemic. I think nobody will forget this. And you know that for the COVID pandemic, during the COVID pandemic and afterwards, there were a lot of studies on why this group of patients has severe disease and the other group of patients has not severe disease. Why are people dying and the others are not dying? And there was a lot of studies called the GWAS studies, Genome-Wide Association studies. And a big surprise came with a SNP that was identified in one of the introns of this BBS17 gene. And this was described by two very huge groups and replicated by other groups. So then the question is how can we link this to what we know about the cilia? So today the pathogenesis is the significance of this SNP is still not elucidated. I asked a very well-known population geneticist about this, still not elucidated, but there have been some works where they did some editing on these genes to look at this as a quantitative trait, and it was shown with the single nucleus RNA, etc., that perhaps there is a role at the level of the circulation of the cilium at the level of the respiratory tract. Anyway, this was a huge surprise. Maybe there is no direct significance, and I can tell you the patients who are mutated in these genes are still alive. They had COVID and they are doing very well. So I checked this immediately. So anyway, but this is surprising, and this shows, how can we interpret all these results. So to finish my take-home message, so in nature there is no waste. I showed you the history of the primary cilium since 1684. Well, it's not a vestigial organ. On the contrary, it's central to biology in cell cycle development and maintenance of organs. Bardet-Biedl syndrome can be like a kaleidoscope of the function of these of the ciliary proteins in different organs. It's quite emblematic. It's often considered and cited as an emblematic ciliopathy. Ciliopathies, they illustrate the rare disease challenges, the unsolved cases, and today we still have patients who are unsolved, and with whole genome sequencing and the dark side of the genome, I mean all the non-coding side and all the discoveries that are being done, we may find new mechanisms. The mechanism underlying, for instance, ciliopathies is extremely complex, and finding therapy is complex because there are different organs and probably different pathogenesis. The models also are difficult to find, and this is the case for many rare diseases. No model is perfect. There is pros and cons for each model, and the prices also are rising very much. For instance, for the mice, it's getting very complicated to do research in this field. And for broader thoughts, what is really a ciliopathy? This is a big question. To what extent the ciliopathy spectrum should go, and also looking at common diseases, as I just mentioned. And also pushing this a little bit further, maybe therapies that we will find for ciliopathies may help in common diseases and have broader applications. We will see. So to finish with, I would really like, I'm absolutely not alone for all this. There are all the teams in the whole world who are working on the subject. But I would like, first of all, to thank the patients, because we are working for them. They are everyday motor, the patient association, the support for the research, the clinical colleagues, because when you are not in the clinics doing research, you have to take care of the patients. And also we are, as Jean-Louis said, we are working very much on clinical pathways, so there is a lot of work at all levels. The members of the lab, and I hope I've forgotten nobody, and the PhDs who have helped us, and also all the local, national and international collaborations. I don't take the risk of putting all the names, but I know I owe so much to all these people. So thank you very much for your attention.
Discussion or questions or comments? Anybody? I mean, this seems to be so central, this cilia, in so many things in your body. Is it, there's a reason for that? There's some master gene that makes this, and that's why so many things are linked?
I think it's, it's very important. Because what is thought is that the surface of the cilia, the self-surface, is quite limited, but would capture more than maybe of the cytoplasmic surface. This is one hypothesis. But of course, it doesn't recapitulate all the biology, etc. But the major signaling pathways that are linked to cilia is huge. I didn't speak about also the role of cilia in cell cycle, of course. Because if we look at these pathways in oncology, etc., there is also a lot of links there. Just focused on rare diseases. So it was overlooked, but there's not one unique gene for cilia. But what is very interesting is to look at the evolution. I took away these slides because I thought it was too long, but to look at the evolution of the cilia in different species and how it appeared. At the beginning it was the flagellum, then it became a sensory flagella, and then and it evolved. So, and also there are a lot of common genes. For instance, Chlamydomonas we saw, and the gene for polycystic kidney diseases. So there's a lot of conservation during evolution. Yeah, please.
So I have a question about the obesity in these BBS patients. Yeah, do they respond to GLP-1? That would be a better solution.
Yes, so it's a very good question. Actually, on Friday, I'm going to a meeting with endocrinologists, the French Endocrinology Society and Obesity. We will speak about that. So, for instance, probably they would respond. To my knowledge, there has been no trial to prove this. There are already patients that have been prescribed by the general practitioner who has prescribed this. It's very useful in patients who have diabetes because at the beginning it's a drug that is useful for diabetes and obesity. So, for instance, for setmelanotide, the trials have been done in the academic way. We have the proof; it has been statistically proven. There are patients that are responders, others that are not. Of course, all these GLP-1 drugs that are really making the buzz worldwide, it's impossible for the patients and the general population not to know about these. It will go, I think it will go wild, that we will not have the answer. We will see by cases. Ideally, and I suppose we will speak about this on Friday with my colleagues, ideally we should do a study. However, most of the patients have been going on the setmelanotide study, which has, actually I haven't been speaking about that, but the setmelanotide has adverse effects. One of the adverse effects is pigmentation of the skin. So they get, because it's the MC4 receptor of the melanotin, so they get very much pigmented. So we have really to follow them on the dermatological side. So of course it would be ideal to do a study on the GLP-1 and Bardet-Biedl. But probably there will be an effect.
Right. And then very interesting.
Thank you.
Extremely complicated for me.
I hope not.
But you mentioned in one of your first slides, Lodenhoek, a Dutch scientist.
Yes.
It turns out that he is the person who discovered spermine and spermidine and human semen.
Yeah.
And he discovered this as a phosphate salt of these polyamines. And then later on, Christian Oriol-Lodi in Collège de France, and they had a collaboration with Jean-Marie Lane, they showed that these polygenic polyamines, they play a very important role in cell division, and in particular in the polymerization of actin. So I wonder if the cell division, polymerization of actin, the actin-myosin complexes does not play a role in proliferation of this kind of disease.
Yes, it's possible, of course, and I think there are quite a lot of. You mean also extracellular matrix that can have a role, and this is something that is, actually under investigation by various labs, because we always look at the cell, but there is the environment, and this also, of course,
And some people, they showed that prior to appearance of a tumor that you can see, there is an enhancement of the polygenic polyamines, POO.
Okay.
And they even thought that they could use this as a therapeutic approach.
Okay.
And then they invented DFFO.
Mhm.
And
Okay.
things.
But what is amazing is these scientists, they really discovered so many things. I mean, it's really stunning. I mean, I didn't get into the details, but this person was amazing.
Thanks. I have a very short question. Very out of curiosity, you showed the list of the different genes that are affected, and one of them is ARL6, and it's only in the Réunion Island. And I was curious because there the Réunion Island before the 17th century was not inhabited. So the question is, this mutation didn't appear in the last 400 years, or was it imported by one of the population that...
I don't know the origin exact of the mutation, but what I know is that it was actually Mrs. Gouron and Jean Muller who did this study, because they found what we call a founder effect, meaning that in La Réunion they have the same mutation in this gene, which comes from a common ancestor. I think they have been dating. It was possible to date with the haplotypes, but to know if it was the first, if it was imported or if it occurred 400 years, that is very difficult to say. But this brings me to another comment. For instance, in Newfoundland, there was a very active group in Bar-des-Bidoles years ago. And it was always thought, we were thinking in Newfoundland there would be one gene because there would be the same, a founder effect. And absolutely not. There is all the genes you can imagine in this very small population. So it's not always dictated. It's probably also because a lot of prisoners came into this territory. Very compact.
Thanks for the talk. I have a question. It's just, so at the beginning you mentioned that the disease, this syndrome is still, or it used to be traditionally characterized as autosomal recessive.
Yeah.
Given what you've learned, or what we've learned recently about the amount of genes that contribute for the phenotypes and for the clinical manifestations, can we still assume that it's an autosomal recessive?
Thank you very much for this question. I appreciate it particularly because it is definitely an autosomal recessive condition. So the genetic counseling is done like for cystic fibrosis. The parents, they are heterozygote and they have 25% of risk of having a child with Bardet-Biedl syndrome. Why is this question very important? Because when the science paper came out in 2001, genetic counselor in the United States did not perform genetic counseling in Bardet-Biedl patients without having all the gene sequence, and even then they were unsure about the inheritance, meaning that for a number of years a number of families were not able to have family planning because of this. So we stay really, and we have discussed this a lot of times with Jean-Loup, because by chance, when you have 26 genes, you can be heterozygote for one of these 26 genes by chance. And definitely you can have modifiers. What I didn't tell you is we find, for instance, mutations in BBS genes in patients who have only retinal dystrophy and not the other symptoms. So why is this? They have no obesity, they have no polydactyly. We have also patients who have retinal dystrophy that is, I will not get in specialist detail, but inverse types. One who is peripheral and one who is central. Why is this? So we are lucky, and we even have an exceptional case of a patient who has no retinal dystrophy, who is 50 years old, who has BBS mutations, and who has a perfect retina. So for us, it's a gold mine because we will really dissect this patient with organoids, because we have collected the fibroblast, and she has collaborated very nicely to the research. So we want to understand what are genetic or epigenetic factors that act on this expressivity, and these could be targets also for therapy. Yeah, please.
So, but you don't know any case of an homozygous for one of the mutations that has no symptoms?
That has no symptoms? No, not yet. Maybe it will come. We do not have. No, not to my knowledge.
Okay. Yes. Last question.
Beautiful story. Thank you, Helen. About your organoid models, are you able to recapitulate this fantastic morphology of these cells with autosegments?
So we are getting there. I would have hoped that today we could have presented the characterization. We are working with, that's why I mentioned her, Vicky Kalatsis in Montpellier, who is a really very high-level expert on these organoids. And indeed, you can see the structure of the cilia occurring at, I think, 150 days in the progenitor of the photoreceptor cells. And we will see if we see some disorganization at that stage. So hopefully yes.
Merci.